Welcome. In the last session, I came up to discussing nucleosides. In this session, I'm going to discuss about nucleotides. Then we will see the rest of the structure of DNA molecule. So, in the last session, we understood that what is a nucleotide. So, I told you it is a combination of nitrogen base plus pentose sugar plus phosphate, right? So these three together will constitute a nucleotide. So now let us see various types of nucleotides in case of DNA molecule. So here, so first we'll take the nitrogen base 89 plus, we know in case of DNA the pentose sugar present is deoxyribose plus the phosphate as usual. Now this results in the formation of a nucleotide, what we call it okay, deoxyadenosine monophosphate results okay in formation of a nucleotide called deoxyadenosine monophosphate. So in short it is called DAMP, and in another words I'll call it deoxyadenylic acid, so also called deoxyadenylic acid. Now let's see by taking another nitrogen base guanine, right, and the deoxyribose remains the same and the phosphate also remains the same, and this results in the formation of deoxyguanosine monophosphate, formation of deoxyguanosine monophosphate. So this in short called DGMP, right? In other words, I'll call it a deoxyguanolic acid. Deoxyguanolic acid. Right. Now the next one will take thymine plus the deoxyribose and the phosphate. Now this results in the formation of a nucleotide called deoxythymidine monophosphate. So results in the formation of deoxythymidine monophosphate. In short, I'll call it DTMP. Right, and in other words, it is called deoxythymidilic acid. So it's called deoxythymidilic acid. The last one, the nitrogen base cytosine. Okay, so the deoxyribose same and the phosphate also same. Then this results in the formation of deoxycytidine monophosphate. So in short, it's called DCMP. Okay, and otherwise we can also call it a deoxycytidilic, called deoxycytidilic acid, right? So these are four nucleotides that we see in case of DNA molecule, right? So till now I was discussing the components of DNA molecule. So now we will see the structure, the whole structure of DNA molecule. So we know that in 1865, when Mendel published his work, we do not know. I mean, in fact, no one at that time know what actually is this DNA molecule. And it was in 1953 only we have come to know the detailed structure of DNA molecule. So let's see the detailed structure of the DNA molecule. So here the detailed structure of DNA molecule was given by two persons, J.D. Watson and F.H.C. Crick, in 1953. So it was given in 1953. So J.D. Watson and F.H.C. Crick, both of them together given the structure of DNA molecule. And the name of the model, what they have proposed, is the double helix model. So J.D. Watson and F.H.C. Crick in 1953 proposed the double helix model of DNA, right? And this structure, that is the double helix model of DNA, it was based on, so this model was based on several observations provided by other scientists, right? So this double helix model was based on several observations provided by other scientists. So the data J.D. Watson and F.H.C. Crick, they took, and they have come to a conclusion that the DNA exists in this double helix model. So here, who are those scientists? Those observations made them come to a conclusion that the DNA molecule exists in the double helix model. So let's see their names. Okay, so here a person named Morris Wilkins. So Morris Wilkins is a British physicist, and another person called Rosalind Franklin, right? So these two persons, they did X-ray diffraction. So X-ray diffraction of that DNA molecule. So these two scientists, that is J.D. Watson and F.H.C. Crick, they took this data, that is X-ray diffraction data of this DNA molecule. So that is one point here. So these two scientists, Morris Wilkins and Rosalind Franklin, they did X-ray diffraction of DNA molecules. Then another person named Erwin Chargaff. So another person named Erwin Chargaff is a biochemist, and from him also they have taken the data. So here Erwin Chargaff, he told that, okay, so it's a base equivalence. We call it now as Chargaff rule of base equivalence. So he has given, okay, a rule of base equivalence. Now what we call it Chargaff rule of base equivalence. Let's see here what Chargaff has given, right? So he told that in a DNA molecule, so that is in double-stranded DNA molecule, okay, in a double-stranded DNA molecule, the sum of purines equals the sum of pyrimidines. So this is what he mentioned, the statement, the sum of purines equals the sum of pyrimidines. So let's take here a double-stranded DNA molecule to understand it. Of course, shortly I'm going to discuss this double-stranded DNA molecule in detail. So here A, G, C, T. Okay, we know that in the last session I told you that the adenine always pairs with the thymine, guanine pairs with cytosine, cytosine pairs with okay, of course, vice versa. Then A here, G, and I told you that adenine pairs with the thymine with 2 hydrogen bonds, and guanine pairs with cytosine with 3 hydrogen bonds. So you can see in this case it's a double-stranded DNA molecule. So I'll take one more. So whatever, you can see the number of purines here. So how many purines we have? Okay, so adenine is a purine and this guanine is also same, right? You can see in this case, okay, 1, 2, 3, 4, okay, 5 and 6. So here the sum of purines, so here the purines are 6 in number. So purines are 6 in number. Now next see about the pyrimidines. So we have here the pyrimidines 1, 2, 3, 4, 5 and this is 6. So you can see in this case there are 6 pyrimidine. So that's what he told, the Chargaff told, that the sum of purines will equal the sum of pyrimidines in a double-stranded DNA molecule. Or you can take the ratio between, so this also you can take it, or ratios between adenine and thymine, okay, and in the same way between guanine and cytosine. So you will get 1, right? So here let's see how many adenine we have in this DNA molecule. 1, 2, 3. Okay, so I'm going to take this first one, the ratios between adenine and thymine. So A by T. So we have here, okay, 3 adenine, 1, 2, 3, and then thymine, when coming to thymine here, okay, 1, 2, 3. So that will be 3 by 3 is 1. So that's what the ratios between adenine and thymine, and also the guanine and cytosine will be constant and they equal 1. So even if you take in this case, okay, G by C, so we'll take even G by C. Let's see, 1, 2, we have here 3. So here 3, and cytosine 1, 2 and 3. That again equals 1. So whatever the ratios between adenine and thymine, okay, or the ratios between guanine and cytosine, they will be constant and equals 1 in a double-stranded DNA molecule. Okay, of course, if it is not double-stranded DNA molecule, we are not going to talk about this Chargaff rule of base equivalence, right? Now let us see the structure of DNA. So I told you that the model of DNA, the structure of DNA, what they have told is that it's a double helix model. Okay, let's see. So this is how we will be seeing a DNA molecule. So it will be having 2 strands. So these are 2 strands, right? Okay, so this is 1 strand and this is another strand. This is one more strand. Okay, so in the double helix model, the DNA molecule have two strands. So it has two strands and helically coiled and helically coiled. So you can see here the DNA molecule is helically coiled, and these two strands are connected by the nitrogen bases. So we'll find the nitrogen bases. So here just I'm giving an overall structure of DNA. So later in detail I'm going to discuss this one. So this is how we see, like, the nitrogen bases that actually bring these two strands together. Okay, now when we see the distance between two strands, so here the distance between two strands will be 2 nanometers. So the distance between two strands is 2 nanometers. Of course you can also tell it in terms of angstroms, then it will be 20 angstroms. Okay, so 2 nanometers or 20 angstroms. So this is distance between two strands, right? And next, when we see here the distance between two base pairs, okay, so here it's the distance between two base pairs, and that will be 0.34 nanometers, or you can also give it in angstroms, then it will be 3.4 angstroms, right? So this is distance between two base pairs. So it will be distance between two base pairs. Now here one coil, right? So one coil is what we call a pitch. So here the one coil is what we say one pitch, and in this pitch you will see 10 nitrogen bases. Okay, so here per pitch there will be 10 base pairs. So there will be 10 base pairs. Okay, right? And say suppose if we take from this point to this point, so even though here I did not mention those many, but we need to think that per one pitch, so that is per one coil, we see there will be 10 base pairs. Okay, so this will be having 10 base pairs, and the distance, the length of this pitch will be, right, 34 nanometers. Angstroms. So it will be 34 angstroms or 3.4 nanometers. So this I'll mention here. So this is length of 10 base pairs. So when we see the 10 length of 10 base pairs, so it will be 3.4 nanometers or 34 angstroms. Okay, so it will be 3.4 nanometers. So here 0.34 is the distance between two successive base pairs, and in a pitch I told you there will be 10 base pairs, and the total length of that 10 base pairs, so from one end to another end you will be having so this range, so that is 3.4 nanometers or 34 angstroms. Next, the two strands are complementary to each other. The two strands are complementary to each other, and they have anti-parallel polarity, and they have anti-parallel, okay, polarity. So when we see here one of the strand, okay, say it has on this side 5 dash, okay, this strand will be ending vector, okay, 3 dash. So I'm going to take a arrow mark in this way, right? And the opposite one will be having 3 dash, and this one will be having 5 dash, and I'm okay putting this arrow mark. So you can see one strand of the DNA is anti-parallel to the other strand. So that is what I have mentioned. So they have anti-parallel polarity. So later I'm going to uncoil this, and we will see in a linear fashion to understand it in detail. So let's see, by uncoiling it, right? So here I'm going to strike it. So here this is one strand, and this is an okay another strand T C. So here, right, so I told you that the two strands are complementary to each other. So what we mean by complementary to each other, so here if you know the sequence of nitrogen bases on one strand, you can predict the sequences of nitrogen bases on the opposite strand. So that is what we call the two strands are complementary to each other. So that's what, so already I have mentioned the nitrogen bases on one strand, so I can come to know what will be the nitrogen bases on the opposite strand. So here it will be G, here A, T, G, G, and this will be A, right? And I told you this will be say suppose 5 dash. So what is this 5 dash? I'll tell you that in detail. So this then the other end of the same strand, it will be having 3 dash. Okay, so this actually called, okay, 5 phosphate end, and this will be 3 OH end. So it will be 3 OH end, and the opposite strand, so I told you, say suppose this is okay running from this 5 dash to 3 dash, okay, the other one will be right in the opposite. So you can see here, so this I'm going to take this as 5 dash and this as 3 dash, and same, this will be the 5th phosphate and this will be 3rd OH end. So how it has come up, the phosphate and okay OH end, that we'll see it shortly. Okay, and we know the nitrogen bases are connected by hydrogen bonds, and I told you in the last session that adenine and thymine, okay, are linked by 2 hydrogen bonds, and cytosine and guanine with 3 hydrogen bonds. So you can see here 3 hydrogen bonds between C and G, and only 2 between A and okay T, right? So now let us see at molecular level. So I have just given okay an idea here. We'll see at molecular level, and one more point before we see the molecular level. Here I'll just mention, so this is okay one strand, and this one is the other strand, right? Right, so I'll see here with the component. So here this is the pentose sugar, so this is the pentose sugar, and here you will see the attachment of nitrogen base. So here I'm just putting up a box. So this is the nitrogen base, and here you will see the attachment of phosphate group. So that is the thing here why I mentioned it a phosphate end. And moreover here we know this is the first carbon, second carbon, this point, this is third carbon, fourth carbon, and this is the fifth carbon. So that is the reason why I mentioned this end as fifth phosphate end. Okay, so it's a 5 dash, so that is fifth carbon end here, which is attached to phosphate. Then you have to keep in mind always that, okay, to the first carbon attached will be the nitrogen base. The first carbon of pentose sugar always attaches to nitrogen base. Okay, so first carbon of pentose sugar always attaches to nitrogen base. So it always attaches to nitrogen base. So now let's see, we know that this pentose sugar, say the pentose sugar, I told you it is attached to nitrogen base. So say here it is 89. So nitrogen base, the pentose sugar, and phosphate. So this all three get together, they constitute one nucleotide. The next one, let's see the next nucleotide here, and here again I am mentioning, okay, nitrogen base, say suppose G, and here the third end of one pentose sugar and fifth end of another pentose sugar, they join to form here, okay, of course, to the phosphate group. So here the third end of pentose sugar and fifth end of, okay, pentose sugar, always they attach to the phosphate group. So next point is that the 5 dash and 3 dash, okay, ends always attach to phosphate, always attached to phosphate. So this is pentose sugar. Again I will mention here, this is the first carbon, second, third, fourth, and this is the fifth carbon. So this is another nucleotide. I'm going to take two more nucleotides that makes us easy to understand. So here T, and this is phosphate, and phosphate. So you can see on this end you will find the third, this is the third carbon, so that's what here OH is present, so that's why it's called okay, 3OH end. It's called 3OH end. Now, so this is, okay, one strand. So here that I have mentioned as a line, but here I have shown at molecular level with the phosphate and pentose sugar and the nitrogen base. So now, right, this phosphate and pentose sugar both together, they just form sugar phosphate backbone, right? So this I'll mention here. Okay, so this one and this one. So this will be a sugar phosphate backbone. So this is what I call sugar phosphate backbone. So it's called sugar phosphate backbone, right? Now we understood that how is this 5 dash and 3 dash. Now coming to the opposite strand. So in case of opposite strand, so let's see here. So while taking the opposite strand, we will be we need to see that, okay, the pentose sugar as inverted. Okay, so here instead of this, I'll just mention stride. Okay, right, so this is sugar phosphate backbone. Now let's see the opposite strand. So I'm going to draw. So here just I have taken so that the two nitrogen bases come face to face. Now let's see, I told you that the nitrogen base always attaches at the first carbon. Okay, so here for that reason I need to take in this way the pentose sugar in the inverted, okay, form. So this is the pentose sugar, and we know here the third carbon. So here this is the first carbon, second carbon, third carbon, fourth, and here we have the fifth one. So this is the fifth one, and we know that at this end we have 3 OH. So that is, okay, third and OH present, and this will be the thymine. We know that adenine pairs with the thymine. Then let's see in the next nucleotide. So you see here again inverted. So this is the pentose sugar, and the third I told you already. So 5 dash and 3 dash ends always attached to phosphate. So here we have phosphate. Okay, this is C, and sugar here. This again attached to phosphate. Right, and here this is the fifth, and you will see again the presence of a phosphate attached. So here this will be the 5 or fifth phosphate, and the opposite will be third OHN. So I hope right now it's clear. So why we have this polarity. So I mentioned one strand from 5 dash to 3 dash and another one in opposite way. Okay, right, and see G, and this is A, and I already told you that the bonds between them will be 2 hydrogen bonds, in this case 3 hydrogen bonds, and in this case, okay, 2 hydrogen bonds. And here again this pentose sugar and the phosphate will form sugar phosphate backbone. So we find there are 2 sugar phosphate backbones in a double stranded DNA molecule. Okay, so here it's clear that I told you the first carbon of the pentose sugar attaches to the nitrogen base. So this is the first carbon. Here also same, first carbon, this is the second, third, fourth and fifth carbon. Right, now let's see other bonds that exist between the pentose sugar and nitrogen base and pentose sugar and phosphate. So here the bond that exists between pentose sugar and nitrogen base. Okay, so between pentose sugar and nitrogen base, the bond present is N glycosidic bond. The bond present is N glycosidic bond. So this is N glycosidic bond, is N glycosidic bond, right? And the bond that exists between the phosphates, okay, so here this is a ester bond and this is again an ester bond. So for that reason, of course, that exists with the phosphate, so I call this as phosphodiester bond. So it's called phosphodiester bond. And already I told you the bonds that exist between two nitrogen bases, if they are adenine and thymine, so there will be 2, and guanine and cytosine 3. So or whatever, all these between nitrogen bases, we will be having hydrogen bonds. So all these are hydrogen bonds that exist between adenine, thymine and guanine and cytosine, right? And now I'll show you a little detail on this. Okay, so here we have the pentose sugar. Okay, and I told you here attached is a nitrogen base. So here I'm going to take a single ring first. Okay, and in this case I'm going to take a thymine. So here we have CH3. Okay, and so this will be NH, and here again there will be N, and here, okay, so we see another double bond O, right? So this is how we see in case of thymine. So this is thymine. Okay, and I just mentioned the numbering here. So this is first, second, third, fourth, and so here we have the fifth one, CH2. So this is the fifth carbon. Okay, and when coming to the numbering here, I'm going to mention again in this case of thymine, this we need to take it as 1, 2, 3, 4, and this is 5. So I hope we remember that the thymine is also called 5-methyl uracil. So thymine is nothing but 5-methyl uracil. So you can see at the fifth point, fifth carbon, so you can see methyl group attached, which I have already mentioned in the last session, right? So this is when we take in case of a pyrimidine, the first carbon of pentose sugar. So this is pentose sugar, the first carbon of the pentose sugar, okay, and the nitrogen of the pyrimidine will form the bond, and that will be the N glycosidic bond. I hope now you understood what that N, okay, in the N glycosidic bond. Now coming to this CH2, so in this case, okay, to this attached is the phosphate. So we find here, okay, right, so the P double bond O here, and then O, and here, okay, I'm sorry, so we have O, P, O, double bond here, and O. So here you see the negative, and in this case also you will see the negative here, okay, right. And this is what I call it an ester bond formation here, okay. So this is in case of, right, thymine. When I have taken that is a pyrimidine, you need to note that the first carbon of the pentose sugar will bond with the, okay, the first position nitrogen in case of pyrimidines. Now let's see, in case if we take purine, okay, so here also same, but in this case, so we find, okay, two rings, and here I'm going to take this as guanine, okay, and here we will be seeing again here N, so that will be the N linkage, and then, okay, so here one more and double bond, okay, this is the first carbon, second carbon, third, fourth, and same we will see CH2, so that will be the fifth one. So here O, okay, P, O, right. So this is how we see, and then, right, so here we have N, and then NH2, okay, and then double bond O. So there we have double bond O, and here NH. Now let's see the numbering. So I'm going to start here from here. 1, this is 2, okay, 3, 4, 5, 6, 7, 8, and 9. So you can see the linkage that will be, I mean the bond formation will be between first carbon of the pentose sugar and okay, the ninth nitrogen in case of purines. So whatever that adenine and guanine, it will be same. You can see that numbering here, okay, between what, okay, the bonding takes place. So between these the ninth one, the nitrogen, and the first carbon of pentose sugar, so there will be bond, okay. And same here, that will be N glycosidic bond. So here also this is N glycosidic bond, okay. right? I hope it's clear. So we have understood so how the bonding takes place and what actually is the ester bond. So this is the ester bond. Hope it's clear now. Here I have drawn another diagram to show you the links. Okay, so here, so this is one ester bond and this is another ester bond, so that will form the phosphodiester bond, right? So this is pentose sugars and you can see the nitrogen bases, and here this will be how we see the okay triple hydrogen bond and double hydrogen bond. So here I'm just mentioning, I'm not able to draw the opposite one. Okay, so here you can see in these two cases it is going to form okay two hydrogen bonds. So same I have taken here this as thymine and this one as guanine. So that's this one is going to form the three bonds with the cytosine, and this one is going to form two hydrogen bonds with the adenine. And you can also see in this case where you find the third OH and this will be the fifth phosphate and right. So this is at molecular level the structure of DNA molecule. Before I conclude, so I have one more point. So the structure what was given by J.D. Watson and F.H.C. Crick was okay, it's a B-DNA. So whatever I discussed till now is about the B-DNA. So later when I discuss about okay objective part of this, I will be discussing even the other types of DNA. We have A DNA, B DNA, C DNA, okay, right? And especially Watson and Crick, they explained about B DNA, and this B DNA will be having right hand helix. So it will be having a it is a right-handed coil, so that's why I mentioned here right hand helix, and it's a B DNA. So whatever I discussed till now is B DNA, right? So with this I will end today's session, so we'll see in the next session.